Okay. Hi, everyone. Thank you. I'm so sorry to be the last person between this session and your lunch. I promise I'll keep it quick and hopefully not so boring. For this project, we worked on the computational approach to autoimmunity risk assessment. During pandemic, it's not in another COVID talk, I promise. During pandemic, we were investigating some of the myocarditis cases in both infection and vaccine. And so we come up with some computational analysis and we automated it so it can be generalized to any kind of autoimmunity related questions. But for this computational framework, we assume that our hypothesis is that if it is an autoimmunity mediated risk, if we can identify some of the proteins, we call it here in mimics, so that we can inform the lab scientists to generate their experiments and hypotheses around it. And so we can help them to prioritize some of the proteins to look into and investigate further. And in my group in Pfizer Drug Safety and R&D, we are a computational team and sitting in the more preclinical space. So we make sure that the drugs are safer before human studies, even animal studies. So we do a lot of omics data analysis and machine learning models, the predictive models for that. Again, if you're interested, I would be happy to talk with you. Initially, we all know the COVID infection. We have a lot of cases worldwide, and we have short-term and long-term effects. And here I'm showing a pathogenesis of, as an example, to more multi-organ failure effects. For each individual, all of us have some underlying factors before the infection or before the vaccination. It could be environment, it could be age, it could be our immune response. And at the time of infection, our responses vary depending on these underlying factors. And at the end, depends, mostly commonly we have short-term effects, like maybe headache or loss of taste or smell, which I suffer a lot from. But also in rare cases, we can have some long-term effects like organ damage or heart complications, including multi-organ failure. So for that, I'm showing here our bottom-up approach to molecular mimicry risk assessment. We started with a pathogenic sequence, in our case, a spike protein, and then we blast the spike protein against every single protein in human proteome. And we are trying to identify if there are any human proteins that are similar to spike protein, but our immune system is attacking the spike protein as well as the human proteins that are similar. With that, we use the blasting algorithm for sequence similarities. In that case, we did two times. One is the 9 amino acid. So later on, we're going to use the immune epitope database, IEDB, for MHC1 predictions. And also, we did the same protein similarity with the 15 amino acid peptides with at least 50% similarity. And then we use IEDB for antigenicity predictions, but also we overlay this data to look at the gene and protein expressions of these peptides, proteins we identify in a tissue or many tissues of interest. It could be hearts, for example, if you're investigating myocarditis, which I'll show the results in a bit. Or it could be multi-organ failures, could be combination of tissues, could be brain, heart, and kidney at the same time. But after that, the sequence similarity is not enough. I think Martin, the keynote speaker yesterday morning, highlighted that the sequences could be not very similar, like 16 to 20% similarity, but the structure could be almost identical. And so we included the protein structure analysis in the last part to our pipeline. So we also look at the spike protein structure and how similar in the human protein the structures in human proteins. And here, what we are covering actually for the autoimmunity could be T-cell mediated or B-cell mediated. And then the peptide sequence search is actually covering the T-cell mediated hypothesis, and then the 3D structure search covering the B-cell mediated hypothesis. For sake of time, I'm skipping the identified potential mimics for multi-organ failures. But in this case, we were looking at 3 tissues at the same time, brain, heart, and kidney. And we're also looking at 3 different variants, alpha, delta, and omicron for SARS-CoV-2. And you can see a lot of peptides identified, a lot of proteins identified. It's about 2,500 proteins that we sift through. Again, we started at the bottom. We have a bunch of, and then we use ranking and filtering for identifying like top 5 or 10 proteins. This is the result for the COVID-19 infection-associated myocarditis. On the left-hand side, we see that the proteins identified with the sequence analysis, which is T-cell mediated potential mimics, and on the right-hand side, which is the structure analysis, again, based on the B-cell mediated. And then the top 3, myosin 6 and 7 and desmoplakin, are the ones that are common in both analyses, that we have higher confidence. If this myocarditis cases happen through autoimmunity, these are the top 3 proteins we have higher confidence that might be the case. If it's not autoimmunity related, then they don't mean much. And overall, in conclusion, we investigated the molecular mimicry hypothesis as a potential pathogenesis of adverse events that are associated with SARS-CoV-2 infection.